So, how is it that we know all of this information about how neurons function? How is it that we know that there's more sodium outside than there is inside a cell, and sodium can leak through its channels, and the sodium potassium pump can deal with that leakage and keep membrane potential in place? Well, the answer is we've used a critically valuable technique called patch clamping. And I want to spend just a minute talking about patch clamping because this is one of those techniques in history that has helped biology advance quite a bit. Just like we talked about the giant squid axon, the patch clamp technique has been vital to our understanding of the functioning of neurons. So take a look at the drawing on this slide. It's showing us how patch clamping works. You can see a drawing of a neuron here, and then you can see this device, this mechanical device sitting above it. There's essentially a needle, a microscopically small needle called a micropipetter. This is all metal, by the way. And then you can see this tube above it, um, referred to as the amplifier, and then there's some tubing that goes off to the side. This tubing is going to apply a gentle suction through this device, and the device is going to be placed down onto the membrane of this neuron. So essentially, it's going to suck the membrane of this neuron up into this needle, just a little bit, just to isolate that little piece of membrane. So they've taken this little section, the section that is being sucked into the needle, and they've blown it up for us. So we're looking at the needle here. Here's the edge of the needle on one side. Here's the edge on the other side. Here's the opening in the middle of the needle. And you can see that there's cell membrane right here, shown in yellow, and there are purple ion channels embedded in this membrane. Here's the inside of the cell. Here's the outside of the cell. So what's essentially happened is this device has sucked just a little bit of the membrane inside the needle, and with it, a couple ion channels, shown in purple again. Now, why would you want to isolate a little piece of membrane? Well, if you get this membrane isolated from the rest of the cell, you can then apply some current. You can apply electrical current down that needle, again at millivolt levels, and you can cause changes to occur in these ion channels. You can cause them to open up, to close. This is how we learned things about these ion channels. When they open, when they close, at what voltage they open, at what voltage they close. And it greatly helped us understand how neurons generate electrical current. So, the ion channels that are embedded in the membrane of cells, all cells, but we're focused now on neurons, they are gated structures. They are not simply open all the time. We know they're specific. Each channel is specific to a certain ion, and we know that they are gated, and something has to happen to cause that gate to swing out of the way and open up the channel. And that thing that has to happen is a change in voltage. Now, when we talk about this voltage, remember, this voltage sits at minus 60 millivolts at rest. So what will have to happen is that voltage will have to change to a very particular millivolt level in order to cause the gates that close off these ion channels to swing out of the way and open the channel up. It's all based on voltage, and as we've said, voltage in a living cell is based on the distribution of charge across the membrane, the distribution of the ions, where the gradients are, and what the sum charge is on the inside of the cell versus the outside of the cell. Now, when you look at the different ion channels, the channels for those three particular ions we said were critical in creating resting membrane potential: sodium, potassium, and chloride. Remember, we said protein is the fourth player, but protein is not an ion. So we're going to think about the other three right now. Each one of these has its separate channel. When you look at protein channels, ion channels, they actually have a lot in common. They're structurally very similar, but they're not identical. They couldn't be identical, right? Because if a channel is only going to let sodium through, or only going to let potassium through, or chloride through, it has to be structurally different from the other channels in order to achieve that goal. These ion channels are essentially tubes again, but interestingly, they're not circular inside. They're more rectangular. You can identify walls, flattened walls, 4 of them, on the interior of these ion channels. Now, again, there are some really detailed differences between these channels that we're not going to spend time talking about. For example, the potassium channel is actually made out of 4 subunits. 4 individual polypeptides make up the 4 walls of the channel. Compare that to the sodium channel, which is actually 1 polypeptide that has 4 different areas or domains within it. These are the kind of details that make one channel different from another, but we don't need to focus on these details. Just know that within an ion channel, any of these ion channels, there are these sort of flattened walls forming this rectangular shaped tube, passageway through the channel. Also know that one of the walls is sensitive to a change in voltage. So essentially, one of the walls is controlling the gate. When a voltage change occurs, when there is a change in the distribution of the positive and negative charge across the membrane, one of the walls is going to pick that up through a voltage sensor and going to either open or close the gate as a result. Now, the other thing to know about these ion channels is this. Each of the 4 walls is attached to a structure outside of the channel called an inactivation particle. Now, all this is a piece of that protein. It hangs outside of the channel until something happens that causes it to change shape and block the opening to the channel. So what that means is there are really 2 gates. There's an internal gate on one of the walls, and there's an external gate called an inactivation particle. The difference between those will be clear in just a minute as we talk about these 2 gates in action. Ion channels can achieve 3 different states of function. An ion channel can be opened up, and in the open state, the specific ion associated with that channel can pass through. And remember, it's going to travel through in the direction that moves it down its concentration gradient from the side where the ion is higher through the channel towards the side where the ion is lower in concentration. So open is one of the 3 states. Ion channels can also be closed. the voltage sensitive gate attached to one of the walls can close off the passageway in the channel. Ion channels can also be inactivated. And what that state is when the inactivation particle has blocked the opening of the channel. That blockage is independent of the gate that's inside the channel. So a channel can be closed through the gate in the interior of the channel, or it can be inactivated through the positioning of the inactivation particle. Now, channel inactivation is really important to the functioning of ion channels because channel inactivation allows channels to close rapidly and stay closed despite some changes in voltage. And channel inactivation will keep a channel closed until membrane potential returns to the resting state. So what does all this mean? This means that the gate involved in closing an ion channel is more sensitive to voltage changes than the gate involved in inactivating an ion channel. There are two methodologies for preventing ions to come through. There's closure of the channel, that voltage sensitive closure, and there's inactivation, which will close it and keep it closed until a very particular membrane potential is reached, and that's the resting membrane potential. Take a look at the drawing on this slide. This is a drawing that represents an ion channel. So if you start down at the bottom, you can see this golden area. This represents a membrane, a piece of a membrane, and this structure here represents an ion channel. Now, they've drawn the ion channel in a way that allows us to sort of peek in to the cavity. The darker purple area in here represents the cavity through the channel, the tunnel. And the lighter purple area represents the rest of the protein. So you can see that these ion channels sort of narrow in the middle. Now you really, you can't see the actual walls, but what they've drawn for us are these lines through the channel to show us that this space in here is actually composed of 4 separate walls. That's why they've drawn it like that. Now, take a look up at the top here. This is just a blown up version of this. Now, the other thing you can see in this is this red ball right here attached to what looks like a string. This is the inactivation particle. This is one of them anyway. Remember, each wall has its own. Here's an inactivation particle. It normally hangs down here. It's just part of the protein. It's just part of the big, massive molecule that has folded up three-dimensionally to create this channel. But it normally sits out here. So when something happens, and it's related to voltage again, this is going to change shape. Actually, this is going to change shape and swing that particle right up into the channel to block it and inactivate it temporarily. Now go up here and look at a blown up version of the channel. What they're showing us up at the top is a potassium ion in orange, and notice that it's hydrated. It's surrounded by water molecules. It couldn't possibly get across the membrane without some assistance, without some help from a channel protein. Now, this potassium can pass through this channel if this cavity inside is open. This blue region right here, this represents that gate. And as long as the gate has swung to open up this passageway, potassium can pass through. If this gate were to swing closed and block this passageway, potassium would not be able to come through. And that's what they're showing us over here. We have three states to an ion channel. We have an ion channel that's open. They're showing us that here. The internal gate is allowing passage through the center of the channel. We have closed right here. The internal gate has swung over and blocked the passageway. And then we have inactivated down here. The inactivation particle has blocked one side of the channel. Open, closed, inactivated. So let's take stock of what we've talked about so far. In all cells, including neurons, ions are distributed across the membrane of the cell in a way that creates gradients. At rest, all cells have more sodium outside than inside. All cells have more potassium inside than outside. More chloride outside than inside. More protein inside than outside. And when you add up all those positives and all those negatives on the inside and the outside of a cell, the cell is relatively more negative inside than it is outside. And that creates a resting membrane potential of minus 60 millivolts. This is electrical potential. This is stored energy in the form of charge. That's what makes it electrical. It's in the form of charge. Neurons take advantage of resting membrane potential, as do muscle cells, in that they can very carefully open and close certain ion channels, particularly sodium and potassium. They can open and close those ion channels and inactivate those ion channels in a very certain particular sequence across time to allow the charge associated with these ions to move along the length of their membrane, along the length of their axon in particular, with regards to neurons. When we put electrical potential into movement, we create electrical current. And neurons are using electrical current for signaling purposes, to transmit a signal, a message over distances. Now, I mentioned a few minutes ago that this moving electrical current is in the form of what we call an action potential, or what we shorthand as an AP. Now I have the definition of an action potential right here. It's a change in the electrical potential associated with the passage of an impulse along the membrane of a nerve cell or a muscle cell, a neuron or a muscle cell. A change in electrical potential associated with the passage or the movement of an impulse along the length of the membrane. That's what an action potential is. On this slide, you are looking at an action potential as it appears on a voltage meter. Remember, we can measure these charge differences across the membrane of a cell, just like you can measure them in your own home, in your electrical system. Now, you actually have three different drawings shown to you on this slide. Up at the top, this is all about the stimulation. In the middle, this is about membrane potential in millivolts. And down at the bottom here, we're looking at what the sodium channels are doing across time. So for all three of these, time is passing by along the x-axis, and there are changing voltages on the y-axis. Remember, what the neuron does, depending on where it lies in the nervous system, is receive a signal from somewhere and then transmit that signal off in another direction. And in order to transmit it, it has to transform it or transduce it into current. So there has to be some signal to start all this, and that's what they're showing us up at the top. We are applying a stimulus. We're applying, in this case, current. Okay? We're stimulating the neuron at its dendrite. Now, remember, all living cells have a resting membrane potential of about -60. And that's where we're starting off on our voltage meter right here. We have a voltage meter applied to the membrane of a neuron right here. And at rest, it comes in right around -60 millivolts. Right here at time 0 is where we're applying our stimulus. The stimulus is taking the current, taking the millivolts in a more positive direction. They're going up, up, up from -60 towards -50. But once they hit right here, right at this point, something very unusual happens. And that something is called an action potential. So the stimulus is applied at time 0. The millivoltage reading becomes more positive until it hits what we call a threshold. And if it hits that threshold, it sets into motion a series of events. Now, those events are going to involve both sodium channels and potassium channels, but they've only drawn the sodium channels for us here. So what's the first thing that happens? While we're at rest, before the stimulus is applied, the sodium channels are closed. Here they're showing us the two walls of the channel. They're touching each other in the middle. That's where the gate is. Here's the inactivation particle. And you'll notice it's hanging down underneath the channel. It's not closing off the channel right now. The channels are closed at rest. The stimulus is applied at time 0. We hit threshold. And what happens at that threshold voltage is that the gate in the middle of that channel senses that voltage, changes shape, and opens. What happens when the sodium channels open? Well, sodium is normally at a higher concentration outside the cell, so it's going to flow down its concentration gradient through this channel towards the inside of the cell, and it's going to take with it positive charge. That's going to make the inside of the cell more positive or less negative. And that's what you're looking at right here. It's becoming less and less and less negative as sodium rushes into that cell. Then we're going to hit this peak. Look where we are. We're way up into the positive range now. It's going to hit a peak. And what's going to happen is the sodium channels are going to close and they're going to inactivate too. That's what they're showing us down here. That's going to stop the sodium from flowing through. Now again, we can't see the potassium channel in this particular drawing, but the potassium channel is going to open up. Potassium is normally higher inside a cell than outside. Potassium is going to rush out of the cell, and it's going to take its positive charge with it. So the inside of the cell is going to become less positive or more negative, and that's what's happening right here. It's dropping right back down into negativity. This is an action potential. It's a change in membrane potential across time. And it's what neurons use to transmit signal. Now, there's one more thing to notice on this slide, and that's the green line on the graph. The green line is put in here just to remind us that there is a threshold that a stimulus has to meet in order to trigger an action potential. If a stimulus is too small, the action potential doesn't occur because the threshold isn't met. So what happens instead is it just peters out. So it is possible, if you think about my earlier example of someone stepping on your foot, it is possible for that event to not even be detected by your nervous system. If the step is small enough and you don't see it and you don't feel it and it causes no pain, your nervous system won't even detect it. That stimulus, that footstep, is simply going to peter right out. No action potential is generated. So we learned a lot about action potentials by studying those giant axons inside of squid. Remember, these axons are so big that you can literally see them in the dish, and we used those for patch clamping studies. It was through those kinds of studies that we understood where the resting potential is in a neuron, and again in all cells, and how the differential opening and closing of these two particular channels could create depolarization and hyperpolarization, and a third state that we haven't talked about yet, which is called repolarization. So depolarization is that event where the sodium channels opened up and sodium rushed into the cell. And suddenly the interior of the cell became much more positive, much less negative. It upset the resting membrane potential. At that threshold moment, that particular millivoltage, the cell membrane becomes alive in terms of its ion channels. We see very rapid and dramatic opening of sodium channels, allowing this depolarization event to occur and starting off the action potential. So when we look at an action potential on a voltage meter, we can see three distinct phases. We can see a depolarization, we can see a repolarization, and we can see a hyperpolarization, or what we commonly refer to as an undershoot. Now, the other thing we're going to talk about with regards to action potentials is this. This system that sends electrical potential into motion, that creates current along the length of a neuron's membrane, is controlled from moving backwards, from sending the signal in the wrong direction through what are called refractory periods. We're going to talk about two types of refractory periods, an absolute refractory period and a relative refractory period. So all action potentials start at that resting state because there is a membrane state that, again, all cells have, which is just their resting state. That's that minus 60 millivolts state. The voltage-gated sodium channels are closed. The voltage-gated potassium channels are closed. We have more sodium outside the cell. We have more potassium inside the cell. We're at rest. Then the neuron is stimulated. And again, it depends on what kind of neuron it is as to what that stimulus would look like. Maybe it's light. Maybe it's pressure. Maybe it's pain. Maybe it's stretch. Maybe it's electricity from another neuron. Something is going to stimulate that neuron. Now, in order to be a large enough stimulus, we have to hit that threshold, and we're going to call it the threshold potential. The threshold potential falls right around, again on average, right around minus 40 millivolts. So that tells us something. That tells us that the stimulus has to be at least 20 millivolts to bring us from minus 60 to minus 40. I apologize, my minus sign moved away from the 60 here. Let me fix that for us right now. In order to get us from minus 60 up to minus 40, we need 20 millivolts of stimulus, at least. Once we hit minus 40, the sodium channels are going to open. That's the voltage that will swing that internal gate out of the way. Sodium will then move down its gradient from the outside of the cell through the channel to the inside of the cell. And it's carrying with it positive charge. So the interior of the cell is going to very rapidly become more positive, less negative. So it will move from about minus 40 at the threshold up to about plus 40, again, on average. Now, at plus 40, the gate inside the sodium channel is going to close. The sodium channels will close. No more sodium can rush in. Then that channel will also become inactivated. The particle will also swing into place. Simultaneously, at plus 40, the potassium channels will open. So the sodium channels are now closed. The potassium channels are going to open. Potassium is normally higher inside the cell, so potassium is going to flow out, carrying its positive charge with it. That's going to make the inside of the cell more negative, less positive, more negative. And this creates what we call repolarization. In depolarization, you're moving more positive away from resting. In repolarization, you're moving back down towards resting. So more positive up away from resting, more negative returning to resting. Depolarization, repolarization.